Thank you. So my job is to introduce really the highlight of well, there are two highlights tonight. One is the introduction of Bruce Beutler, our Texas's most recent Nobel laureate, and the second will be the O'Donnell awardees. So in introducing Bruce, let me just say that we live in a dangerous world. During the several hours that all of us are gathered here tonight in this ballroom, we will all be exchanging millions of bacteria and viruses with each other. And fortunately, nature has endowed us with a strong immune defense system, and hopefully there will be no significant reductions in the guest list. So our immune defense system protects us against all types of microorganisms. It consists of two lines of defense. The first one stops the invaders, and the second one eliminates them. And the first line of defense, which is called innate immunity, constitutes the eyes of the immune system that instantly detects all microbes that invade the body. And scientists have known about innate immunity for more than 100 years, but the molecular basis for its operation was not revealed until 1998, when Bruce Beutler identified a then unique class of receptors on the surface of white blood cells. And these receptors, now called toll receptors, enable us to respond to all types of microbes when we first encounter them. Once activated by foreign microbes, the toll receptors produce various cytokines, which then instruct our second line of immune defense, B cells and T cells, to produce antibodies and killer cells that eliminate the invading microbes. Bruce Beutler's discovery of the toll receptors was done at UT Southwestern during a 12-year period from 1986 to 1998. And as you all know, this monumental work was recognized by the Nobel Prize in Physiology or Medicine this year. Bruce graduated from medical school at the University of Chicago in 1981, after which he moved to Texas, where he did his medical internship and neurology residency at UT Southwestern. He then moved to New York, where he spent 3 years as a postdoctoral fellow at the Rockefeller University, and there he carried out a classic piece of research isolating tumor necrosis factor and establishing its function as a mediator of inflammation. In 1986, Bruce returned to UT Southwestern as a faculty member in the Department of Internal Medicine and as an investigator in the Howard Hughes Medical Institute. Soon after the discovery of toll receptors, Bruce left UT Southwestern in year 2000 to join his father's Department of Experimental Medicine at the Scripps Research Institute in La Jolla. Bruce's father, Ernest Beutler, who recently died, was one of the great hematologists and medical scientists of the last 50 years. Last year, 6 months before the Nobel Prize was announced, we at UT Southwestern convinced Bruce to return to Dallas, where he is continuing his studies of host defense and new studies of mouse models of human disease. It's indeed a pleasure to introduce you to Bruce Beutler, who is Texas's 8th, 14th, or 15th Nobel laureate, depending on how you count the numbers. And I'll have more to say about Nobel numerology later tonight. Bruce is...
Well, thank you all. Thanks very much. It's really a pleasure to be here, and thank you, Joe. I was asked to speak about my own path to the Nobel Prize in 10 minutes or less, and that means compressing about 53 years of life into 10 minutes. I'll try to do that and hit the highlights. I think every one of the Nobel laureates here was asked at one time or another, how does one win a Nobel Prize? And that, of course, usually goes to the question of motive and what motivates each of us, and I think the motivations are all very similar. It's often said that people do their work because of curiosity, and I think that's certainly not sufficient. It's necessary, but not sufficient. A lot of people are just idly curious, and that doesn't get you anywhere. And where medicine is concerned, then certainly there's also a humanitarian motivation, which carries the day for a lot of us. I talked to Sidney Brenner about this very issue not too long ago. We were speaking to some graduate students who posed the question, and Sidney's answer was very close to what mine would be. He said it's to solve problems. And I think we all know the feeling of consonance when you discover something, when you find something that satisfies all the experimental data that you and others have established. That gives everyone a very good feeling. There's also the motivation of being first, which is not to be discounted. People share that, people in science share that with explorers, with great geographical explorers, with mountaineers, with anyone who strives to do something new. So that's certainly also part of the motivation, very legitimate. There's a question that always comes up, do prizes motivate? And since we're talking about the O'Donnell prizes tonight, I think one can say definitely yes, they enrich the experience. And I think all of the lectures I heard this afternoon were just wonderful, and you should all be very proud of yourselves. And I want to congratulate all of you. Nice to have validation so that not only you know you were first or did something important. Now, as to my own story, it's probably very similar to all of yours in some ways. I started off as a child with an intense interest in nature. I think it was just something aesthetic for several years. At some point, maybe in my late preteens or early teens, I began to think about how it is that animals, and all life forms for that matter, organize themselves from inanimate material, and what an amazing thing that is. And how can it possibly occur? How does life seem to command the inanimate world to become living material? The closest word that I could think of for this was molecular biology, and my older brother surprised me with a copy of Watson's Molecular Biology of the Gene, about 1968. And I read through it and was even more fascinated. But I was very lucky. It might have stopped there. It might have been idle curiosity, a little bit satisfied. But I did have a wonderful father who was himself a very distinguished biomedical scientist. And in my early teens, I began working in his laboratory. I learned how to isolate proteins, to purify them to homogeneity, to analyze them, to do first starch gel and then polyacrylamide gel electrophoresis, which was new in those days. And I became quite proficient at these things during high school and during college. And then I kept on in medical school, working in various laboratories. I definitely loved science. My father advised me to go to medical school, which I think was excellent advice, because as we all know, all of us in medicine know, disease always reveals problems with physiology. It can teach us a great deal about how a living system works. And that was the central thing that I learned in medical school. That was what I most enjoyed. I did miss basic science, though, and I missed it during my internship and residency as well. And I went to Mike Brown, who may or may not remember this. I asked him to recommend places that I could go for a postdoctoral fellowship, and he suggested that I might apply to Phil Leader and to Howard Green and probably half a dozen other stellar laboratories. And I wrote to all of them, and I either received no reply or a polite decline. So that was the reason I went to Rockefeller. It was really kind of a matter of chance that I went to Rockefeller, and I worked in Tony Cerami's lab. There, the question at issue was, why is it that infection causes wasting? And for that matter, chronic disease causes wasting and weight loss. And there was an assay system that I developed for a factor that had been named cachectin in that lab. This was an activity made by macrophages in response to lipopolysaccharide, which is a structural component of all gram-negative bacteria. It's known to clinicians to cause fever and shock. and it would induce somehow a factor that would cause fat cells to shut down in their production of lipoprotein lipase. So they would no longer take up exogenous fat. And I purified that factor to homogeneity, and lo and behold, it turned out to be identical to a molecule that others had been working on called tumor necrosis factor. And I knew at that point that this was something strongly induced by LPS. It had, in addition to its ability to kill tumor cells, the property of causing this catabolic state. And I found very quickly thereafter that it was toxic. It would mimic the effects of LPS in large part. And I thought that this was probably a mediator of LPS effects. And it turned out to be so. And it also turned out to be very important in chronic inflammatory disease. And that happened quite early on in my career. I would say it was a mixture of luck and hard work. I then came back to UT Southwestern, and I had a lab of my own. And I set about trying to understand how TNF biosynthesis was controlled. And here I must say I stumbled, I had some false starts, and I had some fairly harsh criticism that was well meant and was very helpful to me. We were looking at questions like, is all of the regulation of TNF transcriptional, or is there translational control as well? And there was. We found that steroids would prevent TNF production, and that accounts for part of their anti-inflammatory effect. But I felt after a while that we were really dancing around the main question, which is how does LPS activate the cell in the first place? Because as I thought about it, this was something that people had known about for 100 years, that LPS existed, that it could activate mammalian cells, and yet we didn't understand how. And for that matter, we didn't really understand how microbes were detected within the first minutes to hours after an infection is established. Nobody knew what the receptors were. And at some point, about in 1993, I decided to take a pure genetic approach to this problem. There had been a spontaneous mutation in mice, actually 2 separate spontaneous mutations that made mice refractory to LPS. And in those days, there were only a handful of markers that one could use to map genes in the mouse. And as the density of these markers increased, I believe there were about 300 of them known in the whole mouse genome when we began, we decided to track down the mutation by positional cloning and find it. Positional cloning is something that nobody does anymore in the way it used to be done because the whole genome sequence is known now. It's a completely different game. And sequencing is millions, literally millions of times faster and cheaper than it used to be. But in those days, it was really very difficult. It always went through 3 stages, each of which was harder than the other 2. And you're supposed to giggle at that a little bit. The 3 stages were genetic mapping, and then one would have to clone all of the DNA between 2 markers on the chromosome. Then you would look for genes, for the gene content, and find the mutation by comparing your mutant to your wild-type strain. And this took about 5 years of concerted effort. And we had bad luck in this process. I would say partly we were hobbled by the enormous size of the region and by the lack of sequencing equipment, infrastructure for sequencing. I looked with envy at all the other successful positional cloning projects going on around me, and I would see that this project involved 2 back clones, and this one involved 3 back clones, and ours had 24 backs covering an enormous interval, and it seemed so unfair to me. But the worst part was that we went through 90% of that region before we found the mutation, and I was getting very scared. And my father, whom I couldn't afford to ignore, was urging me to diversify and give this up and don't put all your eggs in one basket. And I believe I heard similar things from other people, again, very well meant. But I tend to be a very stubborn person, and I just couldn't bear to abandon this project after 3, 4, 5 years of effort. And then all at once, one day, we did find the gene that was a toll-like receptor 4, as Joe has mentioned. And we found the mutation first in one of the strains, then in the other mutant strain, and we knew for sure that we had found the LPS receptor. We knew also that it was part of a family of receptors that turned out to detect almost any kind of microbe. Each one has different specificities for conserved molecules made by microbes. Better still, it turned out that this family of receptor worked in practically all life forms. Jules Hoffman, who shared the prize with me, had encountered toll, the namesake of the family, as a sensor of fungal infection in the fruit fly. Other workers had found quite similar molecules even in plants. And so it's sort of a universal system for alerting the host. I think I'm under 10 minutes still, and I've really told most of the story. After that, I went on and I used ethyl nitrosourea to generate more phenotypes. And for the last 11 years, I've been trying to find almost any molecule relevant to an innate immune response. And then very recently, I've been very happy to come back to Dallas once again, which is where I really had my biggest success, and to set up a lab and then hopefully a center which will compare the immune response in many different multicellular life forms. Now, I put together a list of the main things that I think were important to getting the prize, and because I saw the terrible problem that Rick Perry had at the debate a few weeks ago, I decided to make a list. I would say the first thing is you should have a sincere interest in nature. I mentioned that to you, whatever you do. The second thing, once you've established that, is to choose a good father who can educate you. There is something to having a good shopper's sense and to recognizing an undervalued problem. That I think I did. I think not everyone realized how important it would be to find this mutation. I would say do put all your eggs in one basket. If you have something really important and you believe in it, be stubborn and don't let anyone talk you out of it. And finally, surround yourself with good colleagues on the outside of your group who are critical, because that's important. And also, you have to have some people on the inside of your group who are loyal and faithful and you can persuade to go with you and work with you for a period of 5 years in this case. I would list my current administrative manager, Betsy Layton, in that category because she kept me organized throughout this. I actually did some of the laboratory work too in those days. And Alexander Poltorak, who worked from start to finish on this project, even when it was quite dangerous, let's say. So that's all I have to say. Thanks very much.